what I've been working on, because like other researchers, I like to talk about my work. But I'm also excited for the grad students to learn about state agency jobs and what the kind of work that state agency researchers do. And I see we have another one in the room, or at least two. And so this is our freshwater systems are stressed out by a lot of things. We have big global related issues of climate change. We have more localized issues of eutrophication related to changing land use. We have invasive species, and also these new threats that we're learning more and more about, things like microplastics, PFAS, chloride pollution, so on and so forth, algae toxins. And as part of my job as a research scientist at the DNR, I try to help our management staff understand these effects on our freshwater resources. And so I want to start by telling you about some of the work I'm doing related to invasive species in our large lakes. And I made this map earlier in the week. This is a map of the infested waters in Minnesota. At the time I made this, it was 1,604 water bodies. About 600, a little over 600 were infested by zebra mussels. And I'll talk about the biology of these organisms a little more. And so 68 of them were infested by spiny water flea. And I'm showing these species specifically because they're the focus of some of the work that I'm going to talk about. So the first thing I want to talk about is some of the work that I've been doing in Minnesota's large lakes related to productivity. There's been a bunch of people who've collected data in these systems, dozens of DNR staff. I want to point out this woman, Jody Hirsch. She's a retired DNR taxonomist. And because of her work, we have over 1.6 million individual zooplankton identification and length measurements. So an amazing data set for an invert nerd like me.
Stand the bikes anymore. Oh,
so when I talk about the Minnesota large lakes, what I'm really talking about are these 10 walleye lakes that are really important both ecologically, but also economically and socially. They're so important that DNR Fisheries has a biologist dedicated to each one of those lakes, and they have a monitoring program for not just fish, but also for water quality. And starting in 2012, all of them had a zooplankton monitoring program. The last program started a little earlier in 2006 when zebra mussels were first found in that system. Currently, all of those lakes are infested with either spiny water flea, Bythotrephes, or zebra mussels, or in some cases both. You'll notice Pepin is missing from this map, but it's infested with zebra mussels, and I really won't talk about the data from that system. And so there's lots of ways to talk about food webs, and the way I like to talk about food webs is to talk about energy flow and energy within individual compartments. And so this is a really simplified food web where we have algae being eaten by zooplankton that are eaten by Cisco. And within each food web compartment, I like to think about how much energy is produced, but I also like to think about the connections between food web compartments. You can tell you're like really excited when I said production. This is the most words you're going to see on any slide, and that's because I love this topic so much that I don't want to forget anything. But production, when I talk about production, I'm talking about secondary production. So that's the formation of heterotrophic biomass. And it's a really holistic metric of population success because it incorporates things like abundance, biomass, birth, survival, fecundity, mortality, just pretty much any metric of success that you can think of is somehow included in production estimates. And what is production, like kind of logistically? So in this walleye, it eats food. Some of what it ingests is assimilated. Some of it it's used in respiration, and the waste from respiration is respired out of what's assimilated. Some is turned into biomass, and that process is production. Of course, some of what's assimilated is also lost as waste through feces. So this is a measurement that originated in fisheries to help managers set harvest limits. And when we talk about the unit, so production is actually a flux or a flow, and we express it in units of mass, either by area or by volume through time. And I like it because it's really helpful in understanding what's happening in food webs and changes to food webs. So if we go back to my simple food web and we add zebra mussels, so zebra mussels are mussels from Ponto-Caspian region, and they're pretty small, about the size of your thumbnail, although I've seen them like this big in Lake Superior, where the wastewater effluent comes out. And they're really good, efficient filter feeders, and they can get really high densities. And because of that, they can filter large volumes of water in short time. And so they filter phytoplankton out of the water column, large amounts of phytoplankton. And so they're competing with the planktivorous or the herbivorous zooplankton. And then that might have repercussions higher up in the food chain. And so I want to show you some data from a lake that has a zebra mussel infestation. These are data from Cass Lake. And so I'm going to show you a couple plots that look like this. And so we're going to have time series, and these are annual production estimates. The different colors represent different taxa, and we have units of production along the y-axis. Zebra mussels were first detected in 2014 in Cass Lake, and really we didn't see an effect, and I wish I had this updated until a few years later. And what we see is that the production, overall production has decreased, but we really didn't lose any biodiversity. It's like we took these bars and just squashed them down. And that's pretty typical of what we're seeing in Minnesota for zooplankton in lakes that are infested with zebra mussels. Now, Bifidrephes, the spiny water flea, also has big impacts on Minnesota lakes where it's invaded, but the way it has those impacts is different. So it's just a hog. It can eat tons and tons of native zooplankton. And so it's directly predating upon our native zooplankton, competing with planktivorous fish, so fish like yellow perch and cisco. And there's some interest, I wanted to remember to tell you that there's some interesting studies that are coming out of Wisconsin now that are showing that there's top-down effects on the phytoplankton when there's Bifidrephes in the system, because they eat so many native zooplankton that phytoplankton are released from herbation or herbivory, and so we'll see increased primary production. And these are data from Rainy Lake, and they're pretty typical of a lake that's been infested by spiny water flea. It's an incomplete data set, but spiny water flea was first detected in 2006, and then by 2007, of course, we don't know what happened here, but we see from 2007 on a really large reduction in zooplankton productivity. We don't just see a reduction in the overall productivity. What we're seeing also is species loss, so changing biodiversity. And that's really because this zooplankton has a limited gape size, and so it prefers the smaller native zooplankton as its prey. And so it's preferentially eating those. So I calculated the production, zooplankton community production in the large lakes that at the time, all of them now have one or both of these invasives, but at the time were invaded by spiny water flea, zebra mussels, or both. And I looked at the percent change in the productivity in the system. And I saw that depending on what the invasion scenario is, either zebra mussels, spiny water flea, or both, that the grazing zooplankton had reductions from about 15 to 80% in their productivity. Some of this variability is probably related to characteristics of the lake. Some of it's probably related to time since invasion. And then there's other things that I'm not thinking of that drives this variability. That reduction in productivity in the herbivorous zooplankton is also seen in the predatory zooplankton as well. And so we're seeing change, and some of that's due to probably predation by a spiny water flea on smaller predatory native zooplankton, and some of it's due to indirect impacts. And I can't remember why I put this slide back in there, except it was probably to tell you that I think that this link is especially interesting because the spiny water flea has been shown in many systems, including the Great Lakes, to just be really an efficient predator. And it is as efficient at, it's as efficient at eating native zooplankton as it actually eats more native zooplankton than Cisco and rainbow smelt in Lake Superior and as alewife in Lake Michigan. So it really, when I say it's a hog, it's really a pig. It eats a lot of zooplankton. And so in Lake Mille Lacs was the first of our large lakes that was infested with spiny water flea. And since we have such a great data set from there, I was able to calculate Bithytrophes spiny water flea production through time. And really, there were a couple individuals found at the end of 2009, but right away in 2010, their production was about average of what we see. We see a little bit of bouncing around, and that's probably related to prey availability, timing of our samples, all sorts of things. Because I know the production of the Bithytrophes, I can back calculate how much food they needed to eat to make that biomass. And so what I did was first I picked the production of its favored small cladoceran prey. And so that's what you're seeing here is small zooplankton production through time from 2006. And this story is all, these are data all from Mille Lacs. So 2006 to 2017, we got zebra mussels at this time. Their population really increased in 2011. Bithytrophes, their population increased in 2010. And so what we see is that a pattern where the tiny native zooplankton production doesn't decline right away after spiny water flea infestation, but by 2012 and 2013, we really reduced production of these small zooplankton. So I said I back calculated consumption, and that's what this black line is. And it looks like nothing, right? Like it looks like really small. Here, I think it was like 20%, or sorry, not 20%, very little, less than 10% of the productivity. But then by 2013, it was more than 100% of the small cladoceran production was needed to support spiny water flea biomass. And so how can that be? That's really confusing. And so I think there's a couple of things going on. But first, this is just a different way of looking at those same data. And so I looked at the amount of the small zooplankton production that it took to support spiny water flea. And here's that first year. And then 2013, where we go up to 300%. 100% is about here. So about half the time where they're eating at least 100% or more of the production. And so what's happening, there's a couple things that probably are happening. One, they're probably utilizing food resources that I'm not including in this group, including things that we don't even measure like rotifers. But the other thing that's probably happening is that the way we are doing these calculations is based on the samples we take. And the first sample was taken in May. And so these estimates are from May through September. And we know that zooplankton can their populations can start increasing even under the ice. And so those estimates, that biomass that's produced before our first sampling isn't included in my estimates. And I think that the spiny water flea is dipping into that. And so I think about this like investment, like I know anything about investing, but you put down some amount of principal, right? And then spiny water flea is for sure eating the interest. That's the 100% and lower, but it's dipping into that initial investment as well. So the standing stock biomass that was there, spiny water flea doesn't show up into the system until about July usually. So the zooplankton get a good head start on them. And this has been seen in other systems. These are similar analyses from the other Minnesota large lakes that I had calculated production for at the time. We saw that happening not only in Mille Lacs, but in East Vermilion and Rainy. We see it in the Great Lakes. We didn't see it in West Vermilion yet, but it had only been invaded by spiny water flea for 1 year at that time. And we think that other things are happening because of spiny water flea. And so here's the native zooplankton biomass in September in blue and June in salmon from 2007 on. And spiny water flea showed up in 2010. And we saw that fall biomass for our native zooplankton decreased after that. Spiny water flea's around in September, and so that's not surprising. But what we were a little surprised about is that we're seeing June natives, the biomass of our native zooplankton in June is actually decreasing as well. And what we think is happening is that spiny water flea is reducing the population so much in the fall that when it comes time for sexual reproduction for the zooplankton to make overwintering eggs, the zooplankton are having a problem finding a mate. And so we're seeing that as a kind of a hangover effect into the next year. So I didn't talk really about zebra mussels as much as I've talked about spiny water flea. And this is a picture from the bottom of Lake Mille Lacs. But look at how cute the little crayfish is. And I put this in the slideshow because I wanted to tell you that even after zebra mussels invaded the system, we saw very little change in water clarity in Lake Mille Lacs. And so Don Brandstrader and I, Don is a professor at UMD, we came up with an idea to look at water clarity through time in Lake Mille Lacs. And so these are all Mille Lacs data. Again, this was recently published. And so we looked at water clarity. We took all the citizen monitoring data that we could get. We looked at water clarity from 2000 to 2018. And so during that time, we had a couple invasions happen. In 2006, zebra mussels were first found. And then in 2010, the population of spiny water flea really took off. And so this gray middle line really represents, and this is day of year, so the seasonal pattern in clarity in Lake Mille Lacs historically before, from 2000 on, before zebra mussels or spiny water flea were found in the system. Then zebra mussels came in and we saw that there was increased clarity in the summer. And then when spiny water flea showed up, we saw that there's actually a little bit of increased clarity in the spring, but then by the summer, the clarity pattern follows this historical trend. It happens to be that the time spiny water flea shows up is right around here as well. And so we thought that there was some sort of a top-down effect of spiny water flea happening here. And so this was just a graph to remind us that the zebra mussel population really increased in 2011. The Bithytrophes, the spiny water flea population increased in 2010, the year before, so almost at the same time. And now, instead of just one of those invasive species in a lake, we have both of them, and we have multiple impacts. So we have zebra mussels filtering algae out of the water column, competing with our native zooplankton. We have our native zooplankton community being preyed upon by this really efficient predator. And so the link between algae and zooplankton, our native zooplankton, has really decreased because the amount of zooplankton has decreased. These two groups of organisms do kind of the same job, right? They eat algae, so they're and they're both filter feeders of a sort. And so really they're in competition with each other. And what we found happened was that really the decrease in filtering from the native zooplankton that work was taken up by the zebra mussels. So they basically filled that ecological role. And so total filtering in Lake Mille Lacs is shown in dark blue. Native zooplankton filtering rate is shown in green. And you can see that when their population crashed, their filtering rate crashed. And then zebra mussel filtering is in light blue here. And so basically, we've had a total replacement of function by those two groups. And the net result was very little change in water clarity. I'm trying to remember why I put this slide in here for a specific reason. I think it was to say, it was to say that we often use water clarity as an indicator of ecosystem health. I think we need to be careful about doing that, because here we saw a huge shift of the energetics of the system and no change in water clarity. I think there's the opposite case where we see changes in water clarity that aren't necessarily related to good things like zebra mussels. So that said, over the long term, so I just told you a story that was from here on, 2000 on, but over the longer term, the water clarity in Lake Mille Lacs has changed. There was an increase in water clarity in the mid to late 90s. Usually when we talk about that, we talk about the fact that the Clean Water Act was working, and this is what we're seeing. But this new paper shows that really Mille Lacs isn't special. It's special, but there are a lot of lakes in Minnesota that are having increased clarity. And so this is from Hansen, and they found, and I don't remember, hundreds of lakes that some lakes were getting clear, some lakes were getting less clear. The more clear is in blue here, and so the ones that are getting less clear are in brown. And here's a frequency histogram of that. So if the change is negative, that means it's getting less clear. If it's positive, more clear. A lot more lakes are clear. And so that's something that's happening in Minnesota in general. There's a lot of things that are going on. For sure, the Clean Water Act is part of it, but also work from in Midwest lakes are showing that things like water temperature impacts clarity, lake level impacts clarity, zebra mussels impact. Did you filter zebra mussels out of this data set? No. So zebra mussels impact clarity. And we care about that in fisheries because clarity is part of the story when we talk about water or walleye habitat. I can't believe I forgot the name of walleye. And so there's lots of ways to talk about fish habitat, right? But one way that we talk about walleye habitat is to talk about the thermal optical habitat. And so thermal heat, optical light. And so there's preferred conditions. Walleye have this specialized eye for low light conditions. And so they like to be deep enough in the lake where there's some optimal light conditions and not too warm and not too cold. Sometimes we get anoxia at the bottom of lakes or really cold water. And so they're restricted to some band of preferred, they're not restricted, but this is their preferred habitat. We know they venture out of their preferred habitat. If lakes are getting clear, that means light is getting down further, that preferred habitat is shrinking. And that's not good news if you're a walleye, especially if you're a walleye that could get eaten by another walleye. And so this is some more new work out of the Hansen lab. This is Malum et al. From 2022. What they did was they looked at thermal optical habitat in Minnesota lakes versus catch per unit effort. These are fisheries data, so these are probably gillnet data. And they looked at the relationship between walleye catch and the thermal optical habitat. They found that for both small and medium walleye, as the amount of habitat increased, the catch in the fishery surveys has increased. That relationship didn't hold for large walleye. So there's this relationship that we're seeing in statewide data in thermal optical habitat and walleye population size. And so this is just a reminder that we're talking about thermal optical habitat, and these are clarity data from Lake Mille Lacs, and we saw this increase in clarity in the mid to late 90s. At about that same time here, mid to late 90s, we saw a decline in the number of fish in Lake Mille Lacs. And these are modeled data. I am showing you the age 3 to 6 walleye data because I use the same data set to calculate production. Before age 3, we're not confident that we're doing a good job sampling them. And so I'm not including younger fish. And then after age 6, I think there were just so many data gaps. We chose not, because we were catching fewer older fish, that there were data gaps. And so we didn't include it in our production estimates. But what we saw for these young fish is that we saw a decline in abundance through time. We still see some really good year classes, you know, years where more fish are produced. And so I was able to use the really great data set from Mille Lacs to calculate walleye production. And what we saw was, because abundance is part of production, we saw then this decline in these same this age group of fish, age 3 to 6. Again, we still see peaks related to good year classes, but overall we see a decline in walleye production in the system. I showed this to Dr. Hansen and she said, well, you should see my estimates of optical habitat in Lake Mille Lacs. And these are those. And so optical habitat in Mille Lacs declined in that same time frame. Here's the thermal habitat and here's the thermal optical. So the combined habitat. And then she also said, you should look at the mode of production per unit of available or optimal habitat. And so that's what this is. This is my production estimate divided then by the amount of the lake bottom that is good optical habitat. And what we saw was really interesting. Again, we're still seeing some peaks related to good year classes, but we don't see a monotonic trend through time. We're not seeing either an increase or decrease in productivity per unit of habitat. And one of the researchers in my work group said, this is like, I liked this analogy. So this is like a garden where the garden has shrunk, but per unit of garden where it's as productive as ever. And so really just the amount of habitat has shrunk or changed, and but per unit of available optimal habitat, the system is as productive as ever. This is a different way of looking at those data. So production here is coded from cold to hot, so high production to low production. The years represent the estimates for those individual years here along the X. And so I looked at a bunch of different optical metrics, optical habitat metrics and temperature metrics for this data set. I found that the variability in the optical habitat it best decided the variability in the data set. And so what we saw was from low to high optical habitat, we saw a gradient of low to high productivity, but then we had this cluster from the early 2000s on where we had really low optical habitat, but still a lot of variability in production. We saw that was best explained by cumulative annual temperature. And so we still get years where we have higher production, but those tend to be cooler years. I was really curious about how Mille Lacs performed relative to other walleye lakes. And so Andrew Ripple, who's a professor at UC Davis, shared his data set with me. It has over 800 walleye estimates from around North America, walleye production estimates. And this is a histogram of the data set he's compiled. About 80% of the estimates are between 0 and 1 kilogram per hectare per year. There's a few more between 1 and 2, but then very few way far out. The ones really far out tend to be ones that are associated with Escanaba Lake in Wisconsin, a really productive walleye lake. So I then overlaid the Lake Mille Lacs production estimates. Remember, I have about 30 some estimates, and that's why my bars are so much bigger. But really the distribution of the data is what we're looking at. And what I can say is that, wow, Mille Lacs has performed really well relative to other walleye lakes. In fact, it was the highest estimate. I think Lake Escanaba actually was a little bit higher, but it's comes out in the wash because of the errors related to our estimates anyway. So it was between 11.5 and 12 kilograms per hectare per year, which is just incredible. In more recent years, walleye production in Mille Lacs has been lower, more comparable to average performing walleye lakes. That's not to say we're not concerned about the decline in walleye production in Lake Mille Lacs, but just gives us a little bit different perspective. So now I want to totally shift gears, but sort of, because I'm still going to talk about habitat and food webs. And this is a project that I've been working on for a couple of years. It's related to this nuisance algae, Didymosphaenia geminata, and it causes huge mats of, it's actually stock material across the bottom of streams, but also in some lakes. And so this is more storytelling. The research team that we've assembled to work on this is pretty large. It includes some algae experts from the Science Museum of Minnesota, a bunch of DNR folks, including Lake Superior and Lake Superior tributary biologists, includes the AIS divers, and then microbial ecologists from UW Oshkosh and UMD. And so if you Google Didymo, this is the picture you'll get. So I'll call it either rock, I'll probably call it Didymo, but you might hear it called rock snot, Didymo, Didymosphaenia. This is in New Zealand, and what you're seeing here is Didymo. It's a single cell diatom, and it has the ability to extrude a stock that's polysaccharide, so it's a sugar, but it can extrude, an individual cell can extrude like a meter of it. We haven't seen that in Minnesota. We haven't seen anything like this in Minnesota. We've seen a centimeter, but when you Google it, you'll get this. And so that's just a heads up. That's not what we're seeing. We're seeing much less dramatic, although they do see it kind of like this in the St. Mary's River on the eastern end of Lake Superior. So Didymo has been reported as an invasive around the world. It's considered native to Lake Superior, actually, and the other Great Lakes. It likes cold, nutrient poor, moving water. So typically you don't find it in lakes except along the shoreline if there's enough wave action, like in Lake Superior. And these are pictures from Lake Superior. And about 15 years ago, one of my collaborators, Dr. Mark Edelin, who works at the Science Museum, he started noticing that there was more and more Didymo covering the nearshore zone along the North Shore. He actually goes out and looks for this. So that he noticed this was occurring. I think that's a real observation, not just by chance. They noticed that not only he and some other diatomists noticed that not only are these mats being formed more frequently, they're also being formed at a larger spatial extent. So all the way from Duluth to the Canadian border. Again, we consider Didymo to be native to Lake Superior. And so, but we don't know why these mats are forming. We don't know if this is a native strain of Didymo responding to changing environmental conditions, or if it's a non-native invasive. Then in 2018, fisheries biologist Detta Grand-Murray noticed that there's Didymo in the Poplar River at Lutsen. And this is what he saw. And I just think that was a really astute observation, that he knew this was Didymo. He had worked in the state of Virginia where Didymo is a problem. And so at that time, then we got the Science Museum on board because they have taxonomic experts that we don't have that expertise at the DNR. And so since then, we've been lucky enough to get funding from multiple sources to do some surveying. And we started our project in 2021. And what we found was in the stream. So the Poplar River is right here, Lutsen. In 2021, we actually didn't find Didymo in the Poplar River. We found it in 6 or 7 other streams. The number of diatom cells has to do with the abundance in this plot or in this graph. And what we did find was that it's much more abundant in the streams than it is in Lake Superior. We did find it in Lake Superior north in Lake and Cook counties, but not in St. Louis County. And then in 2022, we found it again. And we found it in a couple of different streams. Again, we didn't find it in the Poplar River. And we found it all the way down to my neighborhood in Duluth, where that little blue shed is when you're on your way up to the North Shore, right there. Found it all the way down there. And again, like a lot more higher density than in the lake. In the lake, what we're finding is that it's in the wadeable part of the water. And so this is my original hypothesis. And so because I knew a little about the I'm an amateur phycologist at best, a little about the biology of Didymo, I hypothesized that really we're going to get mat formation when we have low nutrients, low temperature, and a lot of light. And we actually like this. We changed a little bit, but we like this because it seems to be that although we don't know, we probably are having low nutrients when we're having mat formation, but it really seems to be related really highly to low stream discharge. So the last couple of summers, we've had very little precipitation in Northeast Minnesota. Streams have been very dry. We've seen these Didymo blooms and Didymo mat formation. Also, it seems that light is a really important part of this story because what we're seeing is that it tends to form where there's a wider floodplain. And then as you go upstream, I don't know if you've walked in those North Shore streams after cleaning your boots, you'll notice that depending on the aspect of the bedrock outcroppings that you may see Didymo on half the stream if the sunlight gets to it, but not on the other half. And so it seems like light is a really important part of the story, which we think is a great thing because we know that by planting trees, we can control at some level light. So the next stage of this work really is to go into some food web and fish habitat descriptions. And so this is Didymo in the Devil Track River, and you can see that it just covers the bottom of the river. This is a stream that has coaster brook trout. So these are a species of concern in Minnesota. They're brook trout that spend their adult life in Lake Superior and spawn in the streams. And so what we're doing is we're doing some description of the habitat. We're also looking at brook trout diet. And then we're taking 1 million samples for other things for Lauren to bring back, right? So this is part of a larger effort related to brook trout in Northeast Minnesota streams. So this is kind of at the macroscopic scale, but at the microscopic scale, this is amazing. So these are individual Didymo cells, and these are as well, these big ones. There's a whole different algae community associated with these diatom mats. And in other systems, there's been a whole different invertebrate community associated with Didymo mats. And that's important because stream trout eat stream insects. It's one of the main parts of their diet. And so it's possible that in our North Shore streams that stream insect populations are being impacted either through loss of habitat or changing diet, and that's impacting then the food available to the brook trout. And this is, to me, this is so exciting because this is a midge. It's a maggot. It's a fly larva, and we have been playing around with ways to look at gut contents of midges to see what they're eating. This midge, these are diatoms, so individual algae in this gut of this midge. This is from the Devil Track River, and what we found so far is when we look at these midge diets is that they're not eating Didymo, they're eating other stuff associated with the Didymo in the streams. And what we're seeing in Lake Superior, we have divers working on this, and it's been really great to have them. What we're seeing in Lake Superior is that the Didymo actually goes out like a meter, so basically where we wade. But what we're seeing is really thick growth of some native algae. This is bulbacete, and it grew in mats on suspected spawning reefs. So we're targeting lake trout spawning habitat, and what we're finding is that there's thick mats of algae on them. And so we're concerned that these mats of algae might interfere with multiple early life stages of lake trout. And so this project is part of a bigger effort to map lake trout spawning habitat in Minnesota waters of Lake Superior. Really, we know very little about that. We know that lake trout will come in really shallow and spawn. In fact, if you go, I guess people dive at the reef that's right in town by Vickers in the fall, and the lake trout congregate, which sounds pretty cool. But with this project, one of the things, we've had several challenges, and one is that Lake Superior is hard to sample. It's not like, the methods I learned to sample freshwaters don't really apply in Lake Superior. But we've been working on developing some new methods. This is a bilge pump that we've been using as a benthic sampler, and it's done a great job at sampling benthic invertebrates. It was actually produced by this guy, Chris Pelvier, who works for the Lake Superior team. We've been also going out and sampling places we think that are good lake trout spawning habitat. So here's one example outside of Grand Marais by the Cut Face River. You'll see this, it's sticking out of the water when you drive up the shore. I think it's called Alligator Rock. We set gill nets there last fall for the first time ever. And of course, I had a meeting the day they picked up the gill net, and they caught a lot of fish, like a lot of fish and a lot of big fish. And they were mostly males. And so we plan to keep exploring these suspected lake trout spawning habitats. And we have some ideas too for, one of the problems that we've had is to actually confirm that fish are spawning at those places, either by the collection of eggs or by the collection of larval, so juvenile fish. Part of the problem is that if you put passive gear out in Lake Superior to collect stuff, you might never get it back. And I didn't. And so I got an ROV now, and what they use for Lake Michigan is using images. They will put a backpack electroshocker, the anode cathode down, extended, and they'll turn it on and they'll take pictures. And so, because how hard could it be to ID a larval fish from a picture, right? So we're going to play around with that this summer, and we're going to actually do some mapping of these habitats because hasn't been done either. So it's kind of like going to the moon. One thing that I want to mention is that we still don't know if the Didymo we're seeing in Minnesota is a native strain behaving poorly, responding to changing environmental conditions, or if it's a non-native strain, an invasive strain. We have samples at the genetics lab here in Kew to be processed. And this is really important because in Minnesota, to be considered an invasive species, it's a legislative process, and you have to, the species has to be non-native and harmful to either the ecosystem, human health, or the economy. And so it's not just that this is a different species we haven't seen, it's actually a legislative process. And once we know that, we'll know how the state can respond, because if it's an invasive species that comes from one program responds versus if it's, and I know that sounds really bureaucratic, it is. Another part of the state agencies would respond. But with this project, this is a project that we've done a lot of outreach with, and I think part of that's that it's really weird and people are noticing it. So we've had a lot of newspaper press, a lot of radio interviews with Joe Friedrichs at the community radio station in Grand Marais, some blogs. We have a whole team assembled that's related to outreach that we like to keep up to date with our findings, that we'll share any resources they want. You know, if they want a fact sheet, we'll make a fact sheet. We work with these folks to identify samples to check for Didymo. And actually, this is us talking to Joe Friedrichs, and I don't look very amused in that picture. He came out to the stream and talked with us. Even though Didymo is not officially an invasive species in Minnesota, we were able to get it added. It's considered invasive right now in the Great Lakes because we don't know. It's better to be cautious or conservative. But we were able to get it added to EdMaps, which is an app that you can use to report invasive species. The Minnesota DNR runs the Minnesota part of that. And so if you report on EdMaps, the diatomists at the Science Museum get an email, and then they can go check it out. And you can also report it on iNaturalist, and then you can link these two databases, so they will get an update that way too. I had a really awesome intern last summer. Her name is Danielle Kubel, and she made some outreach materials for this project. We went through the process of getting them officially turned into DNR publications, but they're coloring sheets. They're in circulation in some of the state parks and in some of the county library systems over by Brainerd. We produced a fact sheet that we shared with partners related to the stream work. We're in the process of doing one for the lake work as well. Part of the joy of working with the Science Museum is they reach a lot of people that the DNR doesn't reach. You don't probably interact with the DNR unless you buy a license or get a permit for something. Lots of people come to the Science Museum. They had a day where they made Dynamo buttons at the Science Museum. They had kids looking at Dynamo under the microscope. Here's Danielle. I am supposed to be mentoring her there. And then we just kids just stop and ask us what we're doing. And so we have a lot of opportunities just one-on-one with kids to talk about our work. And then this project is going to be part of the circulating curriculum at the Science Museum. They visit every single county in Minnesota at least once a year, and they have a unit on water health. And there's going to be a Dynamo component to that unit. And so after hearing that and giving that talk, it can feel really overwhelming, and you can feel really hopeless about what we've been doing to our fresh waters. And so this is actually a picture I took after my run in November, but that's what I wanted to share was something that I heard the first supervisor I had in the fisheries research unit, Pete Jacobson, say. So Pete was a researcher, and he has a really good kind of zoomed out perspective of things. And he's good at looking at the big picture. And he would say, we're lucky because we live in a state that has lakes and streams that still have good water quality and good fisheries, but they're not guaranteed to stay that way. That's not the uplifting part. The uplifting part is that it might seem like it's hard to make a difference with big global issues like climate change, but we are able to make a difference locally. So we can do things like prevent the spread of invasive species and be good land stewards to prevent nutrient enrichment and sediments getting into our waters. And doing those local things can add resilience to our lakes and streams against climate change. And so there is hope. We can do things locally to reduce the amount of future stress on lakes and streams. So that hopefully that's more uplifting than most of the talk. And yeah, I think we have time for questions.
Yeah, definitely, yeah. Yeah, I'll open up the questions here. Anyone in the room, feel free to raise your hand, go ahead and start talking, and then also on the chat too.
Okay.
So how do you tell Dynamo apart from like any other algae? I'm picturing like when I'm walking on rocks or something, like specifically I'm thinking sometimes in the boundary waters where it's like, I'm like grossed out by the feeling in my feet, wanting to have my chacos on, and like, how do I tell? Yeah.
It's
a native.
It's a diatom, so the blooms actually don't look green, they look yellowish to brownish. Okay. And if you feel them, even though it's called rock snot, it doesn't feel slimy. It feels more like wet cotton or wet paper tissue. And so there are other diatoms that can make mats that we have in Minnesota. None that we know have made this thick of mats. So you might find a mat of, the other one that I know of is called Cyclotella. And so there are other, but the texture really is to me. And then when it dies, it'll stay attached for a while, but it loses its pigments, so it'll turn whitish. And so we had a report of toilet paper in Lake Superior. 2 harbors is again overflowing their septic or their city system, and it was they didn't bring that to me, thankfully, but it was just sloughed off Dynamo. Yeah, Amy?
So it's really cool that you can look at awesome. Are they eating, and they're not eating Dynamo? Are they eating different things in places with Dynamo than without Dynamo?
We don't know yet, but we want to know that. And so that's part of this next phase.
Okay.
Yep. Yeah, Ray?
So how are you estimating secondary production?
I use the instantaneous growth rate method because we had such good data for Mille Lacs. It's a dream. And that's not the case for the other lakes. We'll have to use different methods
When you get your G
from size data, from the GillNet size data.
literature.
Oh, no, from the DNR database.
No, but I mean for estimating growth rate.
Oh, I am able to estimate it by looking at the change in size between year classes in the Mille Lacs data set. So I'm actually estimating the growth rate too. Yeah, Mike?
Yeah, I had a question about detection of invasive species. And it was interesting to see your zooplankton information because you actually had historical like zooplankton surveys for these lake systems. And I'm curious, is there anything similar in the streams where we had benthic diatom information? And if, you know, so that's one piece. And the second piece is, when did Dynamo become a hot word in Minnesota fisheries managers' brains that they would see that and report that? Like, how do we know that it's changing?
So the first one is historical diatom information. We have very little in Minnesota. There's been some individual projects in the North Shore. The EPA lab has done some surveys. Individual academic researchers have looked, but really we don't have a, even though this is part of my dream, instead of like sentinel lakes, we don't have sentinel streams. We should. But no, we don't have those data. And I'm curious about the southeast part of the state. We plan on surveying that to look. But Dynamo, where it's native and not forming a mat, it's very rare in its community. And so you could look through hundreds or thousands of cells before finding one. And so it would be easy to miss. But no, we don't have really great data. And then Dynamo became, I'm trying to remember when we first started talking about it. I think Mark Edlund had put it on our radar because he's interested in it and he's found it in other places. And then we've been lucky enough to hire people that came from states that had Dynamo infestations. And there was a model that was produced like maybe 15 years ago that predicted that Northwest Minnesota would be a good habitat for Dynamo. And this was based mostly on temperature, stream temperature. So some of the streams that drain into the Red River in the far Northeast, I was kind of surprised. But the North Shore streams didn't actually show up on that analysis. But it's known to like really low phosphorus below detection, so 2 parts per billion or lower. And there's very few places in the state where we would get that. Yeah, Gretchen?
Yeah, so you mentioned there's a trout component, the coastal part to the Dynamo project. I know that like in New Zealand where it's out of control, it's having impact on salmon population. Can you remind us of what that is? It because it's inedible, or they like don't have enough food, and have you seen any signs of that in the North Shore?
We haven't seen signs of it in the North Shore yet, but we've only looked at diets for one season. But so what Dynamo does is it totally covers the bottom of the stream. And I think one, it just makes the habitat like not favorable for trout, but two, it really screws up the biology of the insects. It's a snarly mess. Like what you find after Dynamo takes over is that you'll find filiform body-shaped animals, so things like midges or worms, things that can move through that really just jumbled up mat. And so the high quality, big fat insects, things like mayflies, caddisfly, and stoneflies, they really don't seem to do well in Didymo at all. And so the same amount of bug biomass is there, but the players are different. And I don't know if the food's even accessible when it's in those mats. Yeah, Ben?
Is Didymo being transported above like the first falls of those natural barriers?
That's an awesome question, and we haven't seen it there yet. We've done some looking, but we haven't seen it. And we're going to expand that this summer, where we're going to look at more of a stream network approach in 4 streams where we do a better network scale survey for Didymo. Yeah.
So are there any controls for Didymo? Like, is it just mechanical harvest? Is it, how do we get rid of it? Or do we not know yet?
So there's been a lot of work done on it because, as Dr. Hansen mentioned, that it's a huge problem in New Zealand. And there's been some work done in trying different treatment techniques in South Dakota, it's in the Black Hills. And what I can say is there's nothing sustainable. What you can do is you can kill it with some sort of herbicide or copper, but that's not something we would do in the Lake Superior watershed. It's too valuable of a resource. So, yeah.
I guess we're looking at like, I guess, a history of like productivity of walleye in Lake Mille Lacs versus other lakes.
Yeah.
Throughout the state and others. What, if any, like impact do you think stocking has to do with the sustained productivity there versus other lakes, or is there?
So in Mille Lacs, very little stocking has taken place. I think we did it, I'm looking at John Hansen because I know he knows this answer, but I think we did it a couple years because we were mandated to do it, but really it's a natural walleye, naturally reproducing walleye population. I don't know about those other lakes that were included in the data set that Andrew Ripple gave me. I think there's probably some stocking in those lakes.
Cool. Thanks.
Yeah. I got one on chat here. So Colleen says, I'm wondering, did she say we can take pictures of potential Didymo if we see something and send the pictures to someone or put them on iNaturalist? So
iNaturalist would be great. I'm gonna, oh, yep, there it is. Either on the EdMaps app or on iNaturalist. A lot of people already have iNaturalist. You can put it on there. If you tag it with Didymo or rock snot,
either one works.
yeah, and then it'll get it. Actually, Mark Edlund, who's the diatom expert that I work with at the Science Museum, he watches this like a hawk. He is on it. He gets an email if any of that pops up. I'm very excited.
Okay, nice. And then I also just have a question too. I know it's kind of a recent thing with Didymo, but do you guys have an indication or idea of how it gets transported, moved around? I mean, technically maybe it was native to Superior, but how do you think it gets?
So there's evidence from work in New Zealand
Yeah.
that can spread from gear,
Okay.
including boots. And so, yeah.
Speaking of which, isn't the legislature, at least at last I heard, has a proposal to ban belt sole?
Yeah.
Wasn't that your request specifically?
Yeah, it was. So we as fisheries employees in the DNR, we get asked once a year about if we have any ideas for legislative initiatives.
Which are rarely heard or listened to.
Yeah, and so and so I provided a suggestion that we have a felt sole wader ban in Minnesota. It's our surrounding states have them. I know it's not popular with fly anglers for safety reasons, but really they're hard to clean, and it's not just Didymo that we could be moving around. We could be moving around pathogens. It seems like a lot of the most destructive invasives, the ones that cause the most harm, are the smallest. And so really, this is really just an effort to reduce the spread of everything among ecosystems. So yes.
I think that's cool.
Yep, it made it to the bill. So it's, yep.
How's that for a positive note?
Yeah.
Well, I haven't gotten all positive feedback from my coworkers.
You'll be nominated for DNR.
Thank you.
I also just know we're past 1, so if anyone does have to head out, feel free, but she is hanging back a little bit after the talk too. So if you have more questions, you want to have a more informal discussion, that's also going to be happening after this. So I know those are things going on too. So. And I don't see anything else on chat. So I'm going to end the Zoom.
Okay.
Like the recording, but then we can have a discussion after that. and lots of thank yous online too.
Oh, nice, thank you.
Thank you.